Once upon a time, there was a nitrogen molecule named Dennis. He lived in a berry in the woods and helped create food for the berry through a process called photosynthesis.
One day, a bear named Tony came along, and when he saw the berry, he ate it. Dennis traveled into the body of Tony because he was eaten too. The DNA was in one of Tony's cells. One day, Tony fell in love with a beautiful bear, and soon they were married and ready to have a baby. First, the chromosomes in the cell were replicated during interphase. Next, each replicated chromosome paired up with its corresponding homologous chromosome, which formed a tetrad by crossing over.
Crossing over is a process in which homologous chromosomes exchange portions of their chromatids.
Then, in metaphase, pairs of homologous chromosomes lined up across the center of the cell.
Then, in anaphase, spindle fibers pulled each homologous chromosome pair toward opposite ends of the cell.
Next, in telophase, the separated chromosomes clustered and a nuclear membrane formed around them.
Finally, in cytokinesis, the two new cells were separated from each other.
The new gamete Dennis was in mixed with Tony's wife's gamete to make a zygote, which would grow to be their baby bear.
The zygote began growing because of a process called mitosis.
Before the cell could go through mitosis, the DNA had to be replicated.
The DNA had two strands which formed a double helix. In the first step of DNA replication, the strands of double helix separated, forming two replication forks.
As each new strand formed, new bases were added, following the rules of base pairing.
The result was two identical DNA molecules, each one containing a new strand and an old strand.
For the cell to grow, it also had to replicate RNA, which can be thought of as a disposable copy of DNA.
The two processes involved with making RNA are transcription and translation.
The first step after the DNA was copied and RNA was made was prophase. Prophase takes the longest out of all the phases of mitosis.
During prophase, the genetic material inside the nucleus condensed, and the chromosomes became visible. Outside the nucleus, a spindle started to form.
The next step was metaphase. During metaphase, the chromosomes lined up across the center of the cell, and spindle fibers connected the centromere of each chromosome to the two poles of the spindle.
The next step was anaphase. During anaphase, the chromosomes separated and moved to opposite ends of the cell.
The last step was telophase. During telophase, the distinct individual chromosomes began to spread out into a tangle of chromatin. The cell finally separated into two during cytokinesis.
Unfortunately, while the DNA was being replicated in interphase, Dennis became part of the mutated amino acid because part of the amino acid was accidentally deleted.
This changed the way the DNA code was read as the baby was growing.
Because it was not read correctly, the mutation caused the baby's eyes to develop incorrectly.
When the baby bear was born, the mutated amino acid caused it to be blind.
What?
His parents named him Ray.
Ray lived a great childhood, but when he became an adult, he could not rely on his mother to feed him anymore.
Ray had a very hard time finding berries and small animals to eat, so he would get hungrier and hungrier every day.
One day he smelled delicious berries and headed towards them. He followed the scent into a giant forest.
He took a wrong turn and got lost in the forest. He became very tired and decided to take a nap.
He never woke up. When Ray decomposed, Dennis was released back into the atmosphere where he would remain for a very long time.
Woohoo! That was just the beginning of Dennis the Molecule's adventure on Earth.
But that is a story for another day.
What's up? Testing 1, 2, 3.
This is H-Dog.
And Q doggy dog dog.
DNA replication.
Deoxyribonucleic acid is made from two strands of double helix.
So then the strands come apart. Nitrogen bases, they start to unzip by DNA helicase.
H2 bonds
Protein, adenine, and thymine H2 bonds between cytosine and guanine. H2 bonds. The latter structure is strongest in phosphodiestylene.